record. There we go, we're recording. Perfect. All right, that's what patience gets us. All right, so now I'm gonna go ahead and share my screen, PowerPoint. All right, so people online, somebody give me some feedback. Can you see the PowerPoint presentation?
Yeah, we can see it.
I can see it.
All right,
Yeah, we can see it.
great. Thank you all. All right, so here's how we're gonna do this. We have a handful of people in the classroom and a handful of you online. So if you're online and you have a question, please unmute yourselves. We can all hear you in the classroom and then we'll respond. We're gonna meet here. So this is KCC 144, either on, well, we're gonna meet Monday through Thursday and just to clear the air. So to keep half capacity, you're allowed to come either on Monday and Wednesday or Tuesday, Thursday, okay? And we'll see how this attendance goes. If nobody attends, then we can get rid of this distinction that you can show up 4 days a week, because some people will only attend online. But for this week, let's just say that you're gonna stick to your assigned Monday, Wednesday, or Tuesday, Thursday assignment. Sound good? Cool. These lectures are gonna take anywhere from an hour to an hour and a half. This is a 100-minute, so hour 40-minute class. Probably not gonna take the whole time every day. It's a lot. Today's lecture is about an hour with intro included. So we'll get out about a half hour early. Sound good? Yeah, I mean, we only have so much attention. You all can't pay attention for 2 hours anyway, right? It's not gonna be useful. And then you gotta go to lab, you all gotta eat lunch. So I'll be available the whole time that we have class. So class ends at 11:30. You will be able to reach me at least until 11:30 and then through email always. Cool? So this is my Spalding email, and you all can just call me by my first name. I go by Nick. I don't have my PhD yet. I'm doing my PhD at U of L. Your lab instructor, Nate Steffens, he's also doing his PhD at U of L in my lab. So we're really tight, we're good friends. You can ask either of us for anything. So let's just start out, how we're gonna deal with the pandemic. We're in the midst of a COVID-19 pandemic. So I mentioned you can come to class up to twice a week. At this point, can I go ahead and see your all's campus clear? I know we got a couple of them. Just flash it up for me. So if you do attend in person, we're gonna have the Campus Clear app to say that you're not showing symptoms. Perfect, good. Good, so this way, if you come to class, you're gonna keep me and your classmates safe. Perfect, perfect. All right, so we're gonna wear a mask. We're gonna physically distance ourselves. If you don't, I'm gonna call you out and then you won't do it again. But I will enforce these policies. They're very important. And we're gonna sanitize our areas like I mentioned. So definitely after class, I recommend doing it before class, and then we're gonna wash our hands. You can always attend online. You don't even need to email me. If you just don't wanna come to class on Wednesday, it's raining, you're just not feeling well, just attend online. This is your class, this is your education. And since we just figured it out, all these lectures are going to be recorded so you can go back. And I'll post the PowerPoints as well. We have to do a writing assignment for this course. And historically, the professor who taught this for 40 years just retired. So this is the first time this is being offered from somebody new in 40 years. He had you pick any sort of a disease and explain what bacteria or virus caused that disease. I thought about doing that, but we're in a pandemic. And we're going to focus mostly on bacteria, so you are going to write a paper about COVID-19. This is going to help you learn about the pandemic. Also, we're all going to be nurses, right? No? What are we doing? PT and veterinarian. All right, great. Any sort of healthcare setting, you're going to deal with people who resist. So, writing this paper will give you the tools to combat misinformation in a healthcare setting. You'll be able to tell people why they need to be wearing their mask, why they need to wash their hands before they see their loved ones. So this is going to be good for everybody. And I'll get to learn about your perspective too. So I'll give you in the coming days a rubric for that, but I've got the 5 basic questions that I want you to answer in 3 to 5 pages in the syllabus. So here's how we're going to study for this class. We're going to do a chapter a day. So I recommend that you read that chapter before you come to class. I'm going to be reading the chapters, so I expect you to read the chapters. I'm going to come in and convey the information that's in the chapter on a daily basis, and I'll tell you how long it took. Chapter 1 took me about 2 hours to read. So if you're reading every chapter, do it before you come to class instead of after class. So just get yourself a day ahead over the weekend if you can. As you read, you are college students, but not everybody has the same study skills. So I'm going to assume that you need to learn how to study more, okay? So as you're reading, underline or highlight. This is passive though. So this is better than just reading. It's still not active. So what I'm going to recommend is after you get through the lecture, so you read the book, you come to class, you attend, and then after we end, before you come into class the next day, do a one-page summary. So I started teaching through tutoring, and this is a tutoring tip that I recommend. If you can take a blank piece of paper, just a piece of printer paper or lined paper, whatever it is, and digest 30 pages of material on one front page, you will have actively thought through that material and organized it. So you're studying and you're left with a study guide. So if you do this every day, you've got yourselves 8 pages of study instead of 300 for your quiz. Cool? And you're working along the way. It'll keep you sharp, keep you on it. And, you know, if you're confused, you don't know how to do this, or you think you're doing it wrong, bring it to me. We'll talk about it. We'll figure this stuff out. Feel free to share them with your team, whatever. If you all came in together and you want to make day 1, 2, 3 and put them together, do that. Cool? Try to come up with questions as you read. So don't just accept everything that you see. Try and come up with something. Bring that to class. It'll be good for discussion, or just shoot me an email directly. On that, you got to let me know if you're struggling. Otherwise, I can't help you, especially if you're online. I can see you all in person, but if you're online, you're just a blank participant to me at this point during a presentation. I can't see your eyes. And last thing, try and enjoy this. This is a lot of work. We're gonna learn a lot, so let's just try and enjoy this as we can. Whenever you go to lab, that's where you're gonna be like, this is cool. I can smell it. I can see it. I can count them. So the lab will help you enjoy this course a little bit more, I think. So let's just take a look. This is the first half of the syllabus. As you can see, I've split it up into weeks. So on Thursdays, that's going to be our quiz and exam day every week. So I'm going to hold you accountable every week so that you don't go 2 weeks without studying. Your quizzes are going to be 20 questions, probably a half hour at the end of the class. And the exams, we'll see that's a little bit down here, cut off. We're not going to do anything in class on exam day. Your exam is all that we're going to do on exam day. You're going to have 2 of those, 4 quizzes, and a paper. And that's your whole grade. So the paper is going to be, I believe it was 15%, quizzes are 5% each, and then the exams are 35%. I think the paper is 10% actually. Yeah, so that's a letter grade. And then your quizzes are each worth half of a letter grade total.
I have a question.
All right, so, yeah, let's hear it.
Sorry. You were talking about the summary. Is that a requirement of every chapter that we read? Or is it just,
Nope.
you're saying to study?
It is not a requirement. It is a suggested study skill.
Okay.
I recommend that you do it so that you can digest the material on a daily basis and actively think about the material, come up with questions, but you don't have to submit it. You are not required to do that by any means. Thank you for asking, though.
Okay, thank you. Thanks.
Yep. All right, y'all, let's go ahead and get started. So today is the history of microbiology. We have a lot of dates, a lot of really important things. Unfortunately, we're going to talk about historically important men because women didn't come into the forefront of science until the past couple hundred years. We will talk about at least one very important woman today, though. So let's go back to, let's first get this out of here. There we go. All right, so we started to visualize microbes, so really tiny organisms, after, I believe it was somebody who worked with lenses. He made spectacles. He found that if you took 2 convex lenses, or 2 lenses that curve outwards, and you put them in front of one another, that we could view small things and make them larger. So this was in 1625. Let's get this. Here we go. And so 40 years later, that allowed Robert Hooke to publish Microphagia. And so this led to the discovery of the cell. The cell looked like these really small little rooms, so they named them cells. So then Anton von Leeuwenhoek, shown in this image right here, he used this really rudimentary microscope. So he took lake water and put it on this mount and looked at it, and he saw these little things that were moving. He thought they were little tiny animals, and some of them may have been. They could have been little worms. And this is what he saw. So this is what he drew, and he called these animalcules, very small little animals. So what do you all think some of these might have been? Oh, that kind of looks like a little worm, maybe.
Like parasites.
I'm sorry, what did you say?
I'm sorry, like parasites, bacteria, worms.
Exactly. Really small things. So a parasite, in this class, we're going to describe as a multicellular organism that can infect someone, like a small worm, a small nematode. If you look at H, that looks like it could be maybe bacterial cells in a colony. See, you could have seen some yeast, some fungi. Either way, we saw really small things moving around, and we didn't see that before. So now we want to know, where did this stuff come from? If it's just in lake water, how did it get there? So let's keep in mind, this is the 1600s. There was a group of people called vitalists. This is the big idea of the time. They thought that there was this vital force in the air that allowed for life to spontaneously emerge. But Leeuwenhoek was like, well, I doubt that, because if we have maggots growing on the meat, so decayed flesh, they probably didn't arise from the air. They probably came from some eggs, because that's how flies are born. They're born from eggs, right? And so then there was a scientist named Francesco Redi who did an experiment to test this. So we can blow that up, take a look at this. So this is a very simple experiment. Again, keep in mind this is 1600s. So we had two jars. One did not have a mesh on top, so this is open top. The other had a mesh on top that did not allow flies to encounter the meat. So then he waited a few days, and we only see maggots on the open jar, not on the one with the gauze on top. So, what does this show? Yeah, this suggests that the flies need to encounter the meat to lay the eggs, and that the eggs turn into maggots. Maggots don't just come out of the egg, right? Because we don't see them on this jar. So, if we have our question, was this hypothesis validated? Would you say yes? Yeah. So, if the flies couldn't access the meat, they couldn't lay eggs, so we don't see maggots. Therefore, they did not spontaneously generate. Okay. What was the control in this experiment? So we had one jar with gauze on top and one jar without gauze on top. So we have an experimental and a control. Are you all familiar with controls? Yeah? Okay. So which one was it? Just say it. You're allowed to be wrong.
The control is the one that had the gauze on top.
All right. That is a really good try, but that is going to be the experimental condition. The control is the one without the gauze on top, because Leeuwenhoek said, well, yeah, if the flies can access the meat, then they will lay the eggs. However, if I block the flies from encountering the meat, then they won't lay the eggs, we won't get maggots. So that was the experimental control, or the experimental condition. The control lacked the gauze. So that is one way we could control. Can you all think of another control that we could do if we were to recreate this experiment? So what are we looking for? We're looking for maggots. So another possible control, if we were to reproduce this, would be to just put eggs on the meat. If eggs turn into maggots, you should be able to put the eggs on the meat and then seal it with gauze, and you're still going to get maggots that develop because we know that we put the eggs on there. So that would be a positive control if we were to reproduce this, just to get your heads thinking a little bit. Okay, so why didn't Reddy just seal the jar off completely? Again, think about what the vitalists thought was necessary for life. Yeah, so if vitalism is true, then the flies need the air to survive. So this allowed his critics to accept his hypothesis instead of saying, oh, well, you didn't allow the air in there, so therefore no flies grew. So it was a way for him to deal with his vitalist counterparts. Okay, we're going to go up another 200 years. So this went on for until the 1800s. Louis Pasteur, who's a French chemist, he did a series of experiments to just debunk this once and for all. So let's start at the top. Experiment 1. He took broth. Broth is, you can think of it as bacteria or yeast, so microorganism food. It's a liquid, and you put microorganisms in there, they'll grow. So what he did is he boiled it. That's going to sterilize, kill off everything inside there. And then he either had an open top flask or a sealed flask. The one that was open allowed for organisms to come in, and they grew. Surprise. But if you had it sealed off, nothing grew. So he said, look, life doesn't spontaneously generate. But his counterparts said, well, yeah, because you didn't allow the vital force in. You sealed it off, so therefore nothing grew. So they're like, okay, fine. Let me do another experiment. I'll start with this swan-necked flask. So he did the same thing, boiled his broth, it was sterile, nothing appears, surprise. And then he broke off the top of one of them, that puts us in our original condition, stuff grows, surprise. You let it in, it grows. But nothing grows if the organisms are trapped in the bottom of this neck. So then he tips it over, the broth encounters the organisms, then stuff grows. So only until the organisms get exposed to the broth does something grow in there. So he shows, look, there are things in the air that will grow, but they don't spontaneously emerge. So 1861, Pasteur debunks spontaneous generation. Put that into perspective. That was like 150 years ago, you all. Not that long ago. All right, disease transmission can be interrupted. I'm not sure where these little squiggles came from. Do you have questions so far? We feel good. Cool. All right, so vaccination can prevent against infectious disease. Anybody know how a vaccine works generally? There you go. Yep. So whether it's a virus or a bacterium, if you get a part of something that isn't alive, or in the case of a virus, that's a viable. So if you put a part of something that could hurt you in your body, it mimics a primary exposure. So it's like you got the flu, but you didn't actually get the flu. So that way you build up antibodies, and then when you encounter it again, you could still get sick, but you're going to mount a more efficient, robust response as soon as you get that encounter instead of 2 weeks later. So that's what a vaccine does. And Chinese have been doing this for centuries. I think the book says going back to the 13th or 14th century, they had this idea of variolation, V-A-R-I-O-L-A-T-I-O-N, variolation. So they took smallpox scabs, ground it up, and had people snort them. And then they didn't get smallpox. So in 1796, so almost in the 1800s, Edward Jenner took this into account as he noticed that the milkmaids who were working with cows who had cowpox, so the milkmaids might get cowpox, but then they were resistant to smallpox. Right. So he's like, okay, well, if the Chinese have been doing variolation and these women exposed to cowpox don't get smallpox, maybe I can prevent smallpox by taking a cowpox scab and putting it on a human. So completely unethical experiment in today's world. That's what he did. He took a scab from one of the milkmaids that had cowpox, scratched it into a young boy's arm. That boy was later infected with smallpox. Jenner just straight up gave him smallpox. There was a local response, but the boy survived. So this was the first Western medicine vaccine that was ever used. So the infectious agents were similar, but they weren't the same. But he recognized that he could prevent people from dying from smallpox, which is huge. So let's keep rolling in the 1800s. 1847, Ignaz Semmelweis found that women were dying left and right when they were giving birth. So he noticed that if his physicians, who were almost definitely all men, were going from working with cadavers straight into the maternity ward, that the women were dying from these diseases. I believe they called it childbed fever. But the midwives could help women have children, and their women weren't dying. So what he started doing was making his physicians just wash their hands after they touch dead humans. Before you give birth to before you help a woman give childbirth, wash your hands after you touch a cadaver. All of a sudden, mortality rates drop significantly, but his peers didn't like it, and he was fired from his job. So he saved all these women's lives, but people were resistant, as they still can be today, and he loses his job. Pretty sad stuff. Then right about that same time, John Snow also helps people not die through hygienic measures. So there's a water pump where the local people would go and get their water, but it was, for whatever reason, exposed to sewage. And cholera is a waterborne disease. People were dying from cholera, so he just took out that water pump, and all of a sudden, the disease stopped spreading. So it's hard to keep something clean. You just take it out, and all of a sudden, disease transmission doesn't occur. People don't die from cholera. So both of these show that if we just have some basic measures of hygiene, like not touching sewage or washing your hands after you handle a dead human, that you can prevent other people from getting sick. All right. So to Pasteur. So you all have heard of the germ theory of disease. This is known as the grandfather of germ theory, Louis Pasteur. So he showed that yeasts were able to produce biochemical reactions, that they were alive. So people were drinking wine and they were getting sick. What he showed was that if you seal off a fermentation vessel, so air isn't getting in, that yeast will take sugar and produce alcohol. So he showed that yeast can produce biochemicals. That's big. He also is responsible for a process that we still use today called pasteurization. So people were getting sick from drinking the wine. There could have been bacteria, there could have been molds, fungi, all sorts of things that could make somebody's insides not feel good, much less the alcohol, right? So pasteurization is the low added temperature to something over a long period of time. We still do this to milk. You all go buy orange juice at the grocery store, that's probably been pasteurized. I'm not sure about other food products, but milk, for example, if you boil milk, it'll curdle. But if you pasteurize it, then it's just safe to drink. So this brings us to the controversy of like raw milk at certain markets and stuff. Raw milk is illegal to sell in Kentucky because people can get sick. You have to pasteurize it. You go to California, I think it's different. So germ theory was proposed in 1862, but I'll tell you now, we're still figuring this stuff out. We are still in the age of discovery for germ theory. Okay, so let's go ahead and accept that. So germ theory states that small organisms can make us sick. Small things are alive, they get inside of us, and we get sick as a result. So Joseph Lister, you all have heard this name before. Where have we heard Lister? Listerine, right, mouthwash. So Joseph Lister developed antisepsis, which is, similar to pasteurization. It is the reduction of infectious agents on living tissue. So Lister is a surgeon. You can see here, somebody is getting surgery. And people were getting limbs amputated, but that wasn't why they were dying. They were dying because of these post-operative infections. So you get some sort of a nasty infection, and then that would bring them to death. So what he did, how do we get rid of that? So in 1865, he starts using this spray called carbolic acid, or phenol, Phenol. So phenol was, prior to this, used to clean up like sewage contamination. So it's like, all right, well, if I can clean sewage, I could probably clean living tissue. And it works. To this day, we don't use this because there's better things like alcohol, ethanol, isopropyl alcohol, things that will kill bacteria that are less damaging to tissue. So he would cut off a limb, spray carbolic acid, it got rid of the nasty putrid smell, and people stopped dying from operations. This is a big game changer. All right, moving on to Koch. So Robert Koch came up with something called Koch's postulates. So he studied anthrax, which is a bacillus, a rod-shaped bacterium. And so Koch is valued in microbiology because he found that one organism can cause a certain disease. It's not that you're just breathing in deadly air and get sick and there's just general sickness. Like, no, you can have a disease caused by a pathogen. So let's just start with this figure. So you take this animal, it dies, you can isolate the organisms from, say, the blood or a given tissue, whatever it is. And then you should be able to take that and infect another individual, and that individual will get the same disease that it came from, that the organism died from prior. And that's what he saw, and then you should be able to isolate it out of that organism and subsequently infect it. So this is big information, but in today's world of microbiology, I'm going to urge you all to be careful when you're thinking about Koch's postulates. There are certain examples. So I've studied a lot of the oral microbiome, so bacteria in the mouth. And so gum disease is caused by all sorts of different organisms. You have like 700 different bacteria in your mouth. Different types, not numbers, but types. And they work together to cause gum disease. It's not necessarily that one causes the disease. They work together. So we have to be careful when we say that one organism can cause one disease. A disease like pneumonia can be brought about by all sorts of different organisms. Okay? Moreover, we don't know how to culture every organism that we find. We can find it through DNA, but not know how to grow it because we just haven't figured it out yet. So let's just be careful with Koch's postulates. Okay, so Koch found that one organism can cause a disease. This is important because now we can have what's called pure cultures. So you can isolate this organism and then put it in, we talked about broth, so bacteria food. So he would mix gelatin with the broth, and it would make this kind of like semi-solid where bacteria would grow on top of it. Have you all seen Petri dishes in images? So this is prior to a Petri dish, what people would use. It would be like a thick, semi-solid like gel, essentially. Bacteria would grow on top. And I'll show you an image here in a second how we can isolate, and you all will do this in your lab, I promise. All right, so here's the woman we're going to talk about today. Franny Hess, in 1880, proposed, instead of gelatin, to use agar. So Koch, in his cultures, if he put them in an incubator at like 37 degrees, the gelatin would melt. And that was a problem, because he wanted it to stay solid. Agar does not melt at 37 degrees. We still use this today. Upstairs in your old lab, you're going to use agar in your Petri dishes. Promise. So she was using it in her food, she recommended it, and then Koch was like, that's great. And again, we still use it to this day. So if this is the one woman we're going to talk about, you can expect, I'm going to ask you a question about her. So I'm going to do this throughout the whole semester. I'm going to tell you, oh, that's a good test question. So like y'all should be writing that down or taking a note of it, highlighting it somewhere. I'm going to be asked about her. So this is the procedure known as streak for isolation. This will probably be one of the first things you'll do this week in your lab. So what you can do is you can take a sample and then you dilute it. So it's really heavy here and then less heavy, and then what we're going to get are these little, we're going to call this a colony. So each of these is going to arise from one or a handful of individual bacterial cells. It divides, so all of these are genetically identical. And you can take this one colony and then go infect a mouse or put it in broth, and you'll know exactly what's inside your broth. But if you just in this clump over here, you don't know if you have a red bacteria and a blue bacteria until you isolate them. So that's why we call it streak for isolation. This is a pure culture. An individual colony is pure. How are we doing on time? So good. All right. So then this figure just shows that over time we discover all sorts of new organisms, primarily bacteria. We're going to get some filterable viruses. So there's the tobacco plant, and there was a disease called the tobacco mosaic disease. People took diseased plants, exposed them to a solvent, and then tried to filter it out so that they could capture maybe a bacterium that would cause this disease. But if they took that solvent, so it ran through a filter, see, diseased plants, solvent, run it through, and then take that filtered liquid, put it on another tobacco plant, they still got the disease. So that shows that whatever was causing this disease is small enough to go through a filter. That opens the door for viruses. Viruses are very small, they are not alive. This is how we discovered them. And then just a few years later, 1901, Walter Reed finds that there's a virus also filterable that causes yellow fever in humans. So we're talking like 120 years ago that we discovered viruses cause disease in humans. So the fact that we're able to come up with a vaccine for SARS-CoV-2 in less than a year, just think about the breakthroughs that we've come through. Like, that's really big, really important. Don't take this for granted. All right, keep going. We just have some more dates, some more people. So Anton de Berry in the 1860s found that some fungi can also cause plant diseases. So now we have bacteria, we have viruses, we have fungi. And then in 1880, we discovered that protozoan parasites can cause malaria. So Plasmodium falciparum is the parasite, the protozoa that causes malaria. That's still a problem in the globalized world. And if y'all are interested in malaria, Nate is a bit of a specialist on that. I'm sure he'd be happy to talk to you about it. So then your book has this table with some other famous scientists in microbiology. So let's just take a look. By the way, you don't need to memorize this whole table. This is here just to show you breakthroughs that have happened. Some things that you're going to come across in lab. So Hans Christian Gram. Have you all heard of the Gram stain? Yeah, so things are either Gram positive or Gram negative. It'll be one of the first things you do in lab. Streak for isolation and Gram test. It's basic microbiology. I did this before every single experiment. Not going to even start until I do a Gram stain. So again, we're talking over 100, almost 150 years, and we're still doing the same stuff every single day. Theodore Escherich. You've heard of E. coli. That's named after this guy. Escherichia coli is the genus and species. So he found that infants were getting diarrhea. He described the bacterium. Again, E. coli is still a problem in foodborne illness. Okay, this figure is kind of confusing, but let's talk about it. So generally in microbiology, we have 5 major areas of study. So we have bacteriology, so study of bacteria, study of fungi, mycology, study of multicellular parasites, so your wormy-like organisms, and then we have virology and virus-like agents. So those are the 5 major groups. Then we have these 2 little offshoots down here. So these are less focused on, I guess we could say, than viruses and bacteria. So we have phycology, study of algae, and protozoology, the study of protozoa. So again, protozoa including Plasmodium falciparum that causes malaria. All right, so now we're going to talk about the general groups. So bacteria and archaea. As of now, we think that there's something like 10 million different types. I'm sure there are more, and they're going to keep evolving. There's a lot of diversity in the microbial world, is what I'm trying to get at. Generally, they're going to carry a spherical shape like this. We're going to call these cocci. Again, Nate's going to emphasize this in lab. Or they can be a rod shaped like this. We call these bacilli, bacillus for singular. Or they could be spiral, like a spirochete. They literally look like a spring. And we don't have an image of those, but within prokaryotes, we either have bacteria or archaea. And then I've already mentioned the oral microbiome has about 700 different species. The reason I mention this is to just briefly mention that bacteria exist either in a free-floating, what we're going to call planktonic, so they float like plankton in the water. Well, we don't want that.
Oh, now we're back.
Right. We're back. You can see it?
Yes.
Yep.
All right, great. Thank you. Again, feel free to chime in, y'all. If anything like that happens, just say something, because otherwise it's hard for me to tell. So thank you. All right, so let's go back. And then how do I do this? So I need to get out of that. Okay, here we go. Back at it. Whew. Questions so far? You feeling good? Yeah? All right, so let's talk about archaea. So we generally think of archaea as extremophiles. So phile means love. So they love these really extreme conditions. And when I say love these extreme conditions, they don't go to a hot spring because they just love it. They have evolved through natural selection to deal with certain extreme environments. And since other organisms can't, they just capitalize on that niche. So thermophiles love heat. Halophiles love salt. And then acidophiles are going to love really low pH environments, so really acidic environments. Okay, and then I was incredibly surprised to read this in the book. We don't know of any archaea that cause disease. So when I read that, I'm thinking, okay, you're talking about in humans. We don't know of any that cause disease in plants, humans, or other animals. But that doesn't mean that they don't. It just means that we don't know of any that do. I can almost guarantee if you were to look hard enough, you'd find one that can cause disease. But I've looked at a paper just from like 2003 and says same thing. So we still don't think they cause disease. Really surprising. Viruses. We're not going to hit on viruses as much as bacteria in this class, but generally speaking, they're not alive. So bacteria are nucleic acid, either DNA or RNA wrapped in protein, and they require a host. They can't survive on their own. They can withstand destruction, I guess you could say, but they're not alive. They're not cells. They don't have a plasma membrane. And again, a lot of diversity in viruses. As with fungi, these are eukaryotic organisms, so that means they have a nucleus. Bacteria and archaea lack a nucleus. Eukaryotes have one. They also have membrane-bound organelles, things like endoplasmic reticulum. So fungi, can you all think of any that cause human disease? Yep. Well, that would be a wormy parasite, wouldn't it? It could be. I'm not positive, honestly. I'm going to believe you. I'll be skeptical, but I'll believe you. Right, so you said roundworm? Is that ringworm? Ringworm. Okay, gotcha. So any sort of a skin infection, like an athlete's foot, I think is a fungal infection. Women can get yeast infections. So fungi typically live in their food source. So this is, again, why we need to maintain hygiene. And fungi, eukaryotes, have a nucleus. Protists, these are also eukaryotes. So these include algae and the protista. So some protozoa can cause human disease. What's the one that I mentioned? What?
Algae?
Nope. That's fine. Protozoa like what causes malaria? You don't need to know it. His name is Plasmodium falciparum. I'm just trying to get you engaged instead of talk at you for an hour and a half. Okay, so we can see some protista here. Here we have some algae. Some of these are photosynthetic, so they can harness energy from the sun. If you're not photosynthetic, we're going to call you a heterotroph, meaning that you require chemicals from your environment, not the sun, to make energy. Okay, so we can use bacteria for all sorts of things. We can use them to just understand life generally speaking. They grow really fast. We don't need any sort of permission to do an ethical experiment on a bacteria, right? We don't care about the feelings of a bacterium. We have Beetle and Tatum. They found one gene encodes for one enzyme. Again, let's step back and recognize where we are. In today's world, one gene may not necessarily encode for one enzyme. You all may have heard of alternative splicing. Yeah? So one gene could encode for more than one protein. But breakthroughs, they take time to happen. Luria and Bilbrick discovered that bacteria can mutate and get resistance to a viral infection. If a virus infects a bacterium, it has a special name called a bacteriophage. And then Avery, McLeod, and McCarthy found that DNA is the genetic material in cells, which is pretty big. Who discovered the structure of DNA? Yeah, is that a hand? Come on, you all know this. DNA structure. Crick, Watson, and who else? So we have Francis Crick, James Watson, and then Rosalind Franklin, another woman scientist. Franklin's the one who had the X-ray crystal, and then Crick and Watson took that and ran with it and published the paper. But we're going to give Franklin the credit there. So then Crick, the same guy, in 1958, used E. coli and a virus to show that DNA makes up proteins. So we're just piece by piece getting closer to understanding where life comes from, how it generates, and then what DNA is responsible for. So we either have a nucleus or we don't have a nucleus. We're going to divide the microbiological world into those compartments. Oh my gosh. So you're either a eukaryote with a nucleus or a prokaryote without a nucleus. And this shows a eukaryotic cell. This shows a prokaryotic cell. So the DNA is just in a region, but it's within the cytoplasm. In eukaryotes, we have a nuclear membrane that is going to surround the nucleus, and we have a plasma membrane. So the DNA is protected by 2 layers instead of just 1. All bacteria are going to have a plasma membrane. Okay, this is not in your textbook, but this is really cool, and I think it's really important. Anytime I can bring evolution into the picture, I'm going to. So where did a nucleus come from, right? It didn't spontaneously generate. So there's a thing called endosymbiosis theory. Early prokaryotes, their plasma membrane kind of came in and started to envelop around the genetic material, giving us an early nucleus and endoplasmic reticulum. Okay, pretty cool. With life, things happen by chance, and if it works, then it's going to keep going. If it doesn't, that cell will die. So then that cell gets invaded by an aerobic heterotroph, and that cell survives, divides, and now both cells have it. Then there's a second endosymbiosis where a photosynthetic prokaryote also invaded. So here we have the precursors to mitochondria and chloroplasts by one cell invading another cell. And if you don't believe me, all of our mitochondria have circular chromosomes, just like bacteria. Every single one of them. Pretty cool. All right, we are getting close to the end. All right, so the first antibiotic was produced in 1910, so a little bit over 100 years ago. And this was used to cure syphilis. So this is important because it didn't kill syphilis and the person. It was targeted to the organism and kept the individual safe. Go up another couple decades, and we have the discovery of penicillin. So this is from Alexander Fleming. This is his hand-drawn image. So if we have a mold growing, so penicillium, he noticed that the bacteria around had this kind of like halo effect around them. So this shows that the penicillium was releasing something that killed bacteria in a petri dish. So we isolated this and found that the penicillin is an antibiotic. So that was in 1928. 1940, we started getting mass production of antibiotics. Selman Waxman was the one who coined the term antibiotic. Not important, but it's part of the history, so we'll include it. So then, you know, we're talking like 40, 50 years of antibiotic use, and people are like, this is the magic bullet. We're never going to get sick again because we have antibiotics. And then we start recognizing that certain bacterial strains can become resistant. So have you all heard of any antibiotic-resistant organisms? Flu. So the flu is a virus, and viruses mutate. So I wouldn't call that necessarily an antibiotic resistance, but really good try.
I know that, I'm sorry, excuse me. I know that
Yeah.
some forms of MRSA, MRSA are resistant because they're spores.
That's what I was looking for. Great, thank you. So I can't see you. Can you tell me your name, please?
Latoya Johnson. Sorry, I don't know if my video's on or off, but Latoya.
No, it's on. I just have it projected. You said Latoya?
Yes.
Okay, so like Latoya said, MRSA, M-R-S-A, that stands for methicillin-resistant Staphylococcus aureus. So methicillin is the antibiotic. This organism, named Staphylococcus aureus, is resistant to it, so MRSA. And this is really big in hospitals. It's a skin infection. All right, so we have a lot of resistance developing. So what we can try and do is use less antibiotics. Why does that work? Just take a guess.
Your body doesn't build resistance.
Yeah. So if you have some sort of an infection and you take an antibiotic, let's say that you kill 99% of the organisms that cause the disease, but that 1% is left over, those repopulate, and if they withstood the original antibiotic, they're probably going to continue to be resistant. So if we use less antibiotics, we can avoid pushing the evolution of antibiotic-resistant organisms in the future. I'm not saying that antibiotics are bad. I'm just saying let's not use them unless we have to. All right, we are right at the end here. So at the end of the book, or the end of this chapter, they talk about bioterrorism. Remember a handful of years ago, there was that anthrax scare? That might have been 15 years ago. People were getting packages in the mail with anthrax. That's bioterrorism. Or if someone were to intentionally pollute a water supply with cholera, bioterrorism. Really, really nasty, terrible stuff. Climate change. So the Earth is heating at a much more rapid pace than it would be without humans. That's changing the environment, and that can also increase vectors like mosquitoes. So mosquitoes like warm environments. If the world gets warmer, then mosquitoes can travel around different places, and they're carrying protozoa like Plasmodium that can cause malaria. All right, so what we're going to do moving forward is think about how we have evolved to deal with our environment and how the environment has evolved to deal with our response to it, right? This is a coevolutionary process that's never going to stop until all of existence is over. So we're going to use that and try and keep ourselves safe by using evolution and medicine together. And that's a whole field called evolutionary medicine. And so I want to finish with a quote from my PhD advisor. I think this is really powerful. This got me thinking a whole lot when I started my PhD, and I think this is good for you all as healthcare workers. So it says, like many great ideas in biology, the idea implicating infectious causation in chronic diseases, so diseases that last for decades, right? Not just an acute disease. It has far-reaching implications. It's so simple and so significant that one would think it would have been recognized by many and would be the starting point for discussions on the causes of disease, but not yet. That's where we come in. People like me and Nate, we're going to emphasize the importance of evolution in understanding your health. And if you have questions, I would like for you all to bring them up. So here's what we're doing for the rest of the week. Tomorrow we're going to do chapter 2. Wednesday we're doing chapter 3. Thursday we're going to do chapter 4, and then you're going to take a quiz at the end of class. So from 11 to 11:30, we'll have a 20-question, it might be 15, 20 questions. I haven't written it yet, and that'll cover chapters 1 through 4. I'm not going to go through and pick the one thing that I don't think you read out of that little box in the corner. I'm not going to do that to you. I'm going to pick big ideas or things that I've emphasized in class. So go through and think, all right, what are the 5 things that he might ask? If you're going to look for 5, go ahead and look for 8. So when you're doing your one-page summary that I recommend, come up with study questions. Come up with multiple choice questions. And if you think it's a really good one, you can send it to me and I might use it on a quiz or an exam. So that's how I recommend you all study this week moving forward. Anybody have any questions? No? All right. Online people, do you all have any questions?
Yes, I have a question.
Yeah, let's hear it.
So, the quiz, it will be multiple choice or short answer, or like how we can do that?
I'm going to have everything in this whole class be multiple choice for you.
Oh, okay.
Yep. Now, that being said, I can make some kind of tricky multiple choice questions. I'm not going to, again, I'm not on a power trip, okay? I'm here to help you all learn microbiology so that you can work in a healthcare setting and do a really good job. So that's what I'm here for, is to help you have a good career, not give you bad grades. So it will be multiple choice. You'll get your grades back as soon as you complete your exam or your quiz. And then regarding your rescheduling, if you'll just shoot me an email, we'll coordinate and get all that worked out, okay? All right. Anybody else online have questions?
No, I'm good.
All right, great. Well, thank you all for coming in. Shoot me an email if you need anything, and I'll see you tomorrow, okay? Bye, everybody. Good work today.
Bye. Have a good day.
All right, day 1, the book.